Gyrolab solutions for process impurity analysis deliver broad analytical ranges faster and with less hands-on operations, reducing the need for dilutions and reruns. The urgency to bring biotherapeutics to market quickly is greater now than ever. Bioanalysis of culture-related impurities and product titer is essential for process optimization and product characterization, but often creates bottlenecks unless high throughput and efficient methods are implemented to streamline workflows. Gyrolab miniaturized nanoliter-scale immunoassays streamline bioanalytical workflows while reducing sample and reagent requirements. Gyrolab immunoassay solutions for bioprocess analyses of monoclonal antibody and gene therapies provide high-quality data for faster decision-making. The Gyrolab immunoassay platform is based on flow-through affinity columns within microstructures of a CD. Samples and reagents flow through the CD microstructures using centrifugal force and capillary action as the CDs are spun in the Gyrolab platform. Samples are run in parallel with no crosstalk or edge effects. Working at nanoliter scale using flow-through technology offers expanded performance characteristics and capabilities compared to other assay formats, with very low consumption of sample and reagents. The assay format provides a broad dynamic range, minimizing the number of repeats. The assay workflow is automated and integrated using laser-induced fluorescence detection, ensuring high sensitivity, allowing on-column fluorescence profiles for assay troubleshooting and analysis. Control and analysis software can be used remotely for run setup and data analysis. Software designed for 21 CFR compliance ensures that assays can be developed and transferred through to GMP and GLP environments. Gyrolab provides both speed and higher throughput, shortening overall time to data and to decision making. Process-related impurity analysis is an essential regulatory requirement due to the immunogenic potential of impurities. Gyros Protein Technologies has developed robust time and reagent saving solutions for characterization of bioprocess impurities throughout biological production. Gyrolab kits for bioprocess sample analysis of CHO host cell proteins and protein A ligand impurities are ready to run on the Gyrolab platform and produce data in about 1 hour. Gyrolab assay protocols that accelerate assay development are available for PLBL2, insulin, and human transferrin. The Gyrolab CHO-HCP E3G Kit quantifies CHO cell line host cell protein impurities in bioprocess samples. The sandwich immunoassay is run on a Gyrolab BioAffy 1000 HCCD using the industry standard CHO-HCP E3G ELISA antibody from Cygnus Technologies and detects a broad spectrum of CHO-HCPs. The sandwich assay includes the biotinylated anti-HCP3G capture antibody and an Alexa Fluor 647 anti-CHO-HCP detection antibody. The graph on the left shows the broad 4-log working range of the Gyrolab CHO-HCP E3G kit that minimizes sample dilutions, analyzing bioprocess samples with concentrations ranging from approximately 3 nanograms per mil to 8,000 nanograms per mil. The standard curves run in triplicate in 4 runs on 2 instruments and by 2 operators show the high reproducibility. The bar graph on the right indicates that the Gyrolab CHO-HCP E3G kit delivers data that is comparable with ELISA with a broader dynamic range, results within 75 minutes, and higher throughput of up to 960 data points in 1 working day. Protein A, a ligand originating from Staphylococcus aureus and used in purification of IgG, can leach from the chromatography resin and co-elute with the therapeutic antibody product during purification. Measuring protein A directly in bioprocess samples may underestimate the contamination level due to IgG drug product binding to protein A. Acid dissociation of protein A and IgG prior to protein A quantitation is automated using the Gyrolab mixing CD96, as shown in the mixing CD microchannel diagram on the left. The untreated sample is first exposed to a short acid treatment to separate the drug from the protein A ligand in the mixing chamber. Then, a mixture of capture and detection reagent is added to the mixing chamber to build the capture reagent drug detection antibody complex. After a neutralization step, the mixture is added to the streptavidin-coated affinity columns. Comparison with a commercial ELISA kit using acid dissociation is shown on the right, demonstrating that the Gyrolab protein A quantitation was comparable to ELISA, with 2 of the Gyrolab samples below the lower limit of quantitation. Gyrolab saves time, producing 96 data points in 2 hours. For biotherapeutics produced in E. coli bacterial cultures, the Gyrolab E. coli HCP kit has been developed to determine levels of these host cell proteins in bioprocess workflows. This Gyrolab kit is based on reagents from Cygnus Technologies and has a dynamic range of over 3 logs, from 1 to 3,000 nanograms per mil. Shown in the graph on the left, the Gyrolab E. coli HCP results were comparable to manual ELISA, using the same detection and capture antibodies as shown in the Gyrolab assay format on the right. Gyrolab immunoassays only require manual steps of loading samples and reagents at the beginning of the run and produces results in 90 minutes. Phospholipase B-like 2 protein, or PLBL2, is a process-related impurity that may co-purify with the drug substance and cause immunogenic responses in patients. A 3-step sandwich Gyrolab assay protocol is available to determine PLBL2 impurities in bioprocess samples from CHO cell lines. This assay has a broad analytical range of 0.5 nanograms per mil to 1,000 nanograms per mil. Use of this protocol on Gyrolab systems reduce assay development time and streamline workflows. Human insulin and human transferrin are 2 other culture-related impurities that are often analyzed in downstream bioprocessing workflows. The Gyrolab assays for these 2 impurities have a dynamic range of 25 to 15,000 picograms per mil for insulin and 0.1 to 150 nanograms per mil for transferrin. Gyrolab protocols save assay development time, improving project timelines and streamlining workflows. Gyrolab miniaturized nanoliter-scale immunoassays streamline bioanalytical workflows for bioprocess impurity analysis while reducing sample and reagent requirements. Automating impurity analysis with Gyrolab immunoassays streamlines workflows with results within 1 to 2 hours, delivering broad analytical ranges, reducing the need for sample dilutions and reruns, and reducing in-lab operator time to accommodate COVID-19 workplace considerations, making Gyrolab immunoassays the ideal solution for process impurity analyses.